So the final topic in this unit of food microbiology is food preservation. Food preservation is a necessary process simply to make the food last long so that it is having a longer shelf life. So why there is a need for food preservation is just because food gets spoiled. Food spoilage is very common, and it is defined as any change which takes place with respect to the appearance of the food, smell or taste of the foods and food products, which renders it unacceptable to the consumer. You don't feel like eating. Whether it may be due to microbial growth, microorganisms or not. Means, as again, there are three types of spoilages: physical spoilage, chemical spoilage and microbial spoilage, or you can say the biological spoilage. Worms, insects, pests, they may also lodge and lay eggs, and worms may develop in that. That is the biological spoilage. But here we will be focusing on the most common type of food spoilage, which is caused due to microorganisms, that is bacteria, yeasts, molds, fungi, etc. So foods, they fall into one of the three categories, namely perishable foods, semi-perishable foods and non-perishable foods. Perishable foods, they are fresh food items like meat, vegetables, etc. Semi-perishable foods, they include somewhat whose moisture content is on the lower side, like potato, apples, nuts, etc. Whereas the non-perishable foods, they are very dry. Either they are very dry in nature or having more salt or sugar in nature. So this is the basic idea of food spoilage, and food spoilage needs to be countered by food preservation. So how do you define food preservation? Food preservation is prevention of basically the growth of bacteria, fungi or other microorganisms, as well as slowing down the oxidation of fats which may cause rancidity. Even preserving it from rodents, pests, etc. is one way of food preservation. Taking care that the food which is needed, food or the food products, vegetables, fruits, whatever consumable products are there, that need to be preserved so that its shelf life is increased. Many processes have been designed to preserve the food, and time and again with modern technologies, the techniques of food preservation are day by day advancing. Some basic things have to be considered. Low moisture content will give rise to a good food preservation technique. If you are keeping the food in good preservatives, there are various types of chemical and organic preservatives available, which should be, one thing which is important is that it should be a food grade, consumable, and the preservative which is added should not have any side effects or ill effects on the health. So in this presentation today, I have assorted some 25 different types of food preservation techniques, right from the traditional ones which we have been practicing from ages to the modern industrialized techniques I will be just mentioning here. So even if you can remember and later on go through great details, whatever the food preservation techniques interests you, I think that will give you a fair idea of what food preservation technique is all about. So one of the most common type of food preservation technique which has been practiced by humans from ancient times is sun drying. Drying is one of the oldest techniques, and it is used to hamper the decomposition of food products. Food product deterioration can be easily stopped by allowing it to dry. If no instruments are there, like a hot air oven can be used for drying, but for large scale, a simple good open air clean sun drying will suffice. Fruits and vegetables, they are naturally dried. Even fishes, when they are fished from the sea, they are dried in the sun. In places which do not have enough sunlight to dry, they have still houses, means houses which are made up of glass rooftops where the sun allows and drying can be carried out. The second ancient type of food preservation technique is cooling. What does cooling do is it reduces the metabolic rate of microorganisms. enzymes are not produced, and automatically what happens is that the growth is hampered or stopped. Cooling preserves the food by slowing down the growth and reproduction of microorganisms, and the actions of the enzymes which cause the food to rot are stopped. Enzymatic inhibition is there due to lowering of the temperature. Before the era of mechanical cooling using refrigeration techniques, earlier, as you can see here in this picture, here as you can see, a pot or earthen pots are were filled with the food items which needs to be stored, and they were dug in the ground, and just you can add some little water around to maintain the moisture, which will keep it cool. Earthen pots with some clean hygienic conditions need to be maintained, because once the cooling, if it is not proper, then molds may be grown. So ancient techniques had their own what you can say common sense involved in it. If it is not, as you can see here, a pot within a pot is there, and some amount of filling was done, maybe that contained some treated compounds or antimicrobial compounds, and for long durations this can be stored. The third type of preservation technique is freezing. Today we have refrigerators, and it is the most commonly used process commercially as well as domestically. Even in the houses we use refrigerators for preserving a wide variety or range of foods, including the prepared foods. The refrigeration conditions or the sub freezing conditions are very effective. The fourth type, which is very common in households, is boiling, and the fifth you can say heating. Boiling is usually for liquids, and heating is for semi-solid foods and foods. For heating you have microwave ovens today, which very efficiently heat the foods at a very controlled temperature. Boiling, it is 100 degrees centigrade for few minutes. It kills all types of bacteria except spores. Spores are not killed by boiling, but milk, water, etc., liquid broths, brews, soups, they can be preserved by boiling repeatedly. If there is a lag in the time of preparation and consumption, then these techniques needs to be done. Or if foods are used for marketing, where marketing right from the time of the production to the sale and the consumer consuming it, there is a huge gap. The economy of the market depends upon how long you can keep the food good. Food preservation techniques, they are quite efficient because they resist the microbial growth in transportation, in storage conditions, etc. And when a consumer consumes it, he should get it in a good and healthy fashion. So boiling, heating is carried out at temperatures which are sufficient to kill the microorganisms but not destroy the nutritive value of the food. That has to be taken care of. So this all is an art, almost a skill which is needed, so that it is a fine balance of killing the microorganisms but not deteriorating the nutritional value of the food. At the sixth level, again an ancient one, traditional one, sugaring and salting. Sugar and salts, they tend to draw the water outside the cell. As you can see here in the picture, when there is a hypertonic solution of jams, jellies, pickles, etc., what happens is that salting here, pickle is something different, I will come to that, but here you just imagine pickles for salt. Sugar and salt, these initiate plasmolysis. What is plasmolysis? When in a hypertonic solution the cell is placed, the water it rushes in and shrinkage of the cell takes place. That is the layer, it shrinks within the cell, leading to the death of the cell. Plasmolysis is an example of osmosis. So this is one preservation technique, simply putting the foods into high concentration or high, saturated solutions of salt and sugar. Seventh technique is smoking. Smoking is used to increase the shelf life of perishable food items. Just smoke is given to the foods. This is very ancient. Smoked meat is there, smoked vegetables are there, and we have a very popular dish called as dum aloo, dum dena. Dum dena means smoke. Smoke is used. It also gives a typical flavor to the food. This effect is achieved by exposing the food to smoke from burning plant materials like wood, and some other herbs may also be used. Most commonly, the foods which are subjected are meats and fish which have undergone the preliminary process of curing. Then before cooking they are smoked, and it preserves the food in a nice fashion. The eighth type of food preservation is pickling. As I said, adding salt is salting, but pickling is a method of preserving food in an edible antimicrobial liquid. Vinegar is used, some salt, mixture of vinegar, salt, etc., or the extracts of the material which the pickle is made can also be of help here. Chemical pickling, this is chemical pickling, and the food is placed in an edible liquid. Even oils are used, which inhibits or kills the bacteria and other microorganisms. Fermentation, like addition of ginger, turmeric, etc. The pickles, they have all types of herbs, spices, condiments, and they themselves, they become the antimicrobial agents. There is another method called as fermentation pickling. During pickling, fermentation process is allowed to happen, and that produces lactic acid, and that inhibits the growth of another organisms. So a probiotic type of phenomenon takes place. The ninth type of preservation here is sodium hydroxide or lye. It's called as LYE, which makes the food too alkaline for bacterial growth. Bacteria, they are somewhat comfortable in growing in slightly neutral conditions. Plus or minus 0.2 is favorable, but when it is extreme, even extreme acidic conditions may inhibit the growth of organisms, but it does not render good. For extreme acidity is not good, but somewhat alkalinity like around 8.5, which is at a edible level, makes food good. And this sodium hydroxide will saponify the fats. The fats will be saponified, which will change the flavor and texture, but nevertheless give a good preservation life in which the foods are placed in sodium hydroxide. 10th type of preservation technique is canning, which we find in the markets, in the malls, ready-made foods. Canning involves cooking the food, sealing it into sterile cans or jars, and boiling the container so that the microorganisms and other harmful bacteria, they are killed or at least weakened, so that they do not grow inside the cans. Foods, they have varying degree of natural protection against spoilage, and they may require a final step of pressure cooking. Canned foods, they are usually pressure cooked. So canned foods, they, if they harbor spores, then that becomes very dangerous, as we have seen in our earlier lectures. The 11th technique is gelling, use of gels, solidifying agents. Food may be preserved by cooking in a material containing gels. The materials like gelatin, agar, maize flour, corn flour, arrowroot flour, all these gelling agents, they give a natural protection to the food and does not allow the growth of the microorganisms. Some foods, they naturally form a protein gel when cooked, and that gives a preservation. 12th is jugging. This is one method practiced in some countries, is the process of stewing the meat. Okay, meat or fish that is stewed in a covered earthenware. As you can see, it is made up of clay, and it is covered. The meat which is to be jugged is usually cut into pieces and sealed tightly with a brine or gravy. As you can see here, this is a bucket, and inside it the cut meat products are there. It is placed on fire. This is the jug, and it is covered with a earthen lid and allowed to heat slowly for long duration. So smoke is there, heat is there, and inside a jug, so where the heat gets trapped, the meat also get cooked. It is usually for meat and fish products. Jugging, it is called as jugging. 13th technique here is curing. This process draws the moisture from a substance by osmosis. you take the food product and just rub it with salt or sugar. Substances which are food substances which are rubbed with salt, sugar, or a combination of two, what happens is that all the microorganisms which are present on the surface, they are killed by plasmolysis, just as the technique we saw in sugar and salting. Here nitrites and nitrates are also oftenly used to cure the meat so that it gets a characteristic pink color. So this is one of the technique. 14th technique, fermentation, which we all know, such as used to produce cheese, wine, beer, and here specific microorganisms, or in a combination they are used, which combat the spoilage from other microorganisms, those which are not allowed. So the fermentation organisms, they carry out the fermentation, grow in large numbers, and their metabolites and their growth itself does not allow the overgrowth of other organisms, and the only favorable organisms are remained, which preserves the food. Next is what you can say, the fermentation technique used with respect to and metabolites which are being synthesized. Means fermentation microorganisms which are growing, they produce some compounds like bacteriocins or antimicrobial compounds which inhibit the other microorganisms. So they create an environment by themselves or their metabolites, a toxic environment for the growth of other microorganisms. So this is one of the most commonly employed techniques in food industries, where fermented products are formed. Next is pasteurization, a process which is used for preserving liquid food. In this method, milk is heated at about 70 degrees centigrade for 15 to 30 seconds. This is called as flash sterilization, and immediately the bacteria is killed by what you can say, subjecting it to 10 degrees centigrade. So this is a temperature fluctuation which results into killing of the microorganisms. Flash sterilization is immediately followed by cooling at 10 degrees centigrade. So pasteurization followed by cooling, a combination technique. These are techniques used in the industrial level. These are modern techniques and use controlled mechanisms with respect to modern instruments, digital instruments. Next is vacuum packing. Removing the air does not allow the growth of microorganisms. Vacuum packing, it stores the food in a vacuum environment, usually in an airtight bag. A plastic bag is used and its airtight vacuum environment does not allow the bacteria, or it does not allow the bacteria to use the oxygen, because oxygen has been removed and no spoilage due to microorganisms can be observed. Some more modern techniques we will be seeing, that is preservative and food additives, the color compounds, preservatives such as citric acid is there, acetic acid is there, and a huge range of chemicals, pure chemicals, organic chemicals, inorganic chemicals, which are of the food grade, can be used, which are antimicrobial in nature, which inhibit the growth of bacteria or fungi, and including the molds. Or some antioxidants can be added, such as oxygen absorbers, which inhibit the oxidation of food constituents. So these chemicals, which are called as food preservatives or food additives, are very common in packed foods, canned foods, foods which are found, ready-made foods, which we can say. The next technique at the industrial level is use of irradiations. This is also called as cold sterilization. Remember, use of ultraviolet light or radiations for preserving the food is called as cold sterilization. Irradiation of food is exposing the food to ionization radiation, and this kills the bacteria. Treatment effects include killing the bacteria, molds, insects, pests, etc., and reducing the ripening and spoiling of foods. At a higher dose, it induces sterility of the food products. So this is very commonly and effective method in the food industries. Some more advanced techniques involve pulsed electric field or PEF. Electroporation is one of the method for processing cells by means of brief pulses of strong electric field. Small pulsed field is given, and pulsed electric field means not a continuous one but discontinuous one, which holds the potential as a type of low temperature alternative pasteurization for sterilizing the food products. It is as good as pasteurization but at a low temperature, where use of pulsed electric current is there. Next is modifying the environment itself. It is one way to preserve the food by operating it on the atmosphere around. We have air conditioners. What we do when we apply an air conditioner to our house, we modify the environment. We condition the air according to the need. We can regulate the temperature, we can regulate the humidity, the moisture. Similarly, carbon dioxide can be employed. Carbon dioxide, it prevents insects, depending upon its concentration, insects, molds, and also it prevents the oxidation damage of the grains. So a controlled amount of CO2 can be added in the atmosphere. That is a process where we say modifying the atmosphere. Then non-thermal plasma. This is a latest technology which produces a flame of ionized gas molecules. Here you can see this is a flame of ionized gas molecules such as helium or nitrogen, and on the surface this process subjects the surface of the food to this flame of ionized gas molecules, and this causes the microorganisms on the surface to die off, and it is very safe for the composition of the food also. The next method is high pressure food preservation, pressure cooking, which we all know, but industrial pressure cooking is totally different, because the pressure inside the vessel can exert 70,000 pounds per square inch or more. See what happens, how much we can imagine that why this much amount of pressure is needed. High pressure food preservation, or it is also called as Pascalization, which refers to the use of food preservation technique that makes use of high pressures as high as 70,000 psi. You see, don't go on to the figure. I have somehow shown you the figure of a pressure cooker, that is only symbolic, but pressure inside the vessel, high pressure, is used because the quantity of food is more. At the industrial level, you are not having 1 kilo or 2 kilo foods which you put in your pressure cooker. Let us think about food at 100 kilos, 100 tons. For that, huge pressure is required. Food is processed so that it retains its fresh appearance, its flavor should be retained, texture, nutrient should be remained, while disabling the harmful microorganisms from the food. All this can be easily achieved when large amounts of food are subsequently subjected to simultaneously subjected to the high pressure, and this is an industrial technique which is very commonly used in the large scale production of food products which are packed and marketed. Finally, biopreservation. This is one very growing area of modern food preservation technology, which is use of natural or controlled microbiota. That is, you add microorganisms or microorganisms which inhibit other microorganisms as a way of preserving food. So you add consortium, a mixture of microorganisms or antimicrobial compounds or the antimicrobial secreting microorganisms in the food, and they are allowed to grow within. So that is a useful bacteria that itself can become a source of protein. That is, we call it them as the beneficial bacteria or the fermentation bacteria, and their fermentation products which are produced are used for biopreservation to control the spoilage of the food and to render the pathogenic microorganisms inactive. Finally, there is one very interesting technology in vogue today, that is called as hurdle technology. Hurdle technology is a method of ensuring that the pathogens in the food will be eliminated or controlled by combining more than one approach. We have seen 24 approaches. The 25th approach is combining whatever the approach you have just studied into combination and ensuring that you create hurdles for the microorganisms to grow. These approaches can be thought of as a hurdle to the pathogen to overcome so that it will be inactivated. It cannot remain active in the food. So that is a hurdle technology. Whatever suits best, that combination of methods are used. So that's all about food preservation. Remember, preserve and treat your food as you would treat your body, as you preserve your body. You should remember that whatever you eat, remember that that food which you eat, in time that food will become your body. So therefore food preservation is very important. So once again I repeat, listen to this lecture again, pause wherever necessary, read when you have paused, read, don't listen. When you listen, don't read. Listen, don't multitask by reading and listening simultaneously. Do one thing at a time. You have the pause button as a powerful tool with you, and I hope that helps in gaining you a better understanding of food microbiology. So this completes our discussion in this unit about food microbiology. Hope you enjoyed it, gained some insights and knowledge for your degree course in microbiology. Thank you. See you soon.